can be a really good swimmer. Now, that doesn't mean that a bunch of sperm are made on day 1, and then 60 days later, all those sperm are deployed in the form of ejaculate, and then the cycle starts over again. So it's a little different than the ovulatory menstrual cycle. Rather, at any given point in time, like right now, if you have testes, you have some sperm in your testes that are immature and cannot swim, cannot deliver those contents to those genetic contents, rather, to the female egg. And you have some sperm that are mature, and you very likely have some sperm that are so mature that they are dying off or that they're dead. Almost certainly also, regardless of your age, you have some sperm that are healthier than others, are better swimmers than others. This is just the way the system works. Now, the process of spermatogenesis involves a couple of things, but a lot of the players are the same as the process of developing the so-called oocyte, the immature egg. We've got GnRH from the hypothalamus. That's going to be a player. We have FSH, follicle stimulating hormone, although the name's a little bit of a misnomer in the context of spermatogenesis, because in the context of spermatogenesis, there is no follicle. What we're really talking about is FSH for stimulating the maturation of the sperm cell. So not egg follicle, but sperm cell. But we still have GnRH, FSH, LH. And rather than the ovary being the target of those hormones, it's going to be the testes. So most everybody should know that the testes and the ovaries are the so-called gonads. The testes, of course, reside outside of the body. There are instances where the testes fail to descend during development. Certainly, if the testes don't descend on time, that's something that doctors need to be made aware of, the pediatricians be made aware of, because that can prevent fertility. Why would that be? Well, it turns out that the testes reside outside the body in the scrotum because the temperature conditions under which spermatogenesis can occur and under which healthy sperm can be maintained are very restricted and is approximately 2 degrees cooler than the rest of the body. This is very important. I think this is something that used to be discussed a lot more, but isn't discussed so much these days. But keeping the testes cool enough doesn't necessarily mean keeping them cold, although there is a place for using cold exposure, deliberate cold exposure, to improve sperm quality and number, and perhaps even testosterone levels. We'll talk about that a little bit later. But keeping the testes about 2 degrees cooler than the rest of the body is absolutely key. If sperm get too hot, they die. And if spermatocytes, the cells that give rise to sperm, get too warm, well, then oftentimes the sperm that develop are not healthy. Not healthy in a number of ways. Either they can't engage in fast forward progression, and that is swimming, or they will lack the ability to deposit their DNA contents within the egg. So again, whatever is contained in the ejaculate is going to be a mixture of different sperm qualities, and sperm of different ages will impact the quality, but also the temperature under which those sperm developed is going to impact their quality. And so we're going to get into tools a little bit later, as I mentioned, but just to give you a simple takeaway, if you are hoping to conceive in the next 90 days, right? The spermatogenesis cycle takes 60 days, but then the sperm actually have to migrate from the testicle into the so-called epididymis, which is a related structure, and then into the vas deferens, and then into the urethra, where it can be part of the ejaculate. In order for sperm to do all that properly, undergo that maturation, and then exit in ejaculate in a way that's healthy, or that the sperm is healthy. If you plan to conceive children, or to try and conceive children in the next 90 days, you definitely want to avoid exposing your testicles, that is your scrotum, to elevated temperatures. So that means definitely avoiding hot tubs, definitely avoiding hot baths. Now, a brief hot bath or hot tub or hot shower isn't going to be a problem. Although, if you're really interested in conceiving, I would avoid hot tubs and hot baths as much as possible. Hot showers are probably fine. But if you're going to go into a sauna, for instance, you might want to rethink that decision. And if you do decide to, you almost certainly would want to bring a cold pack in that you could hopefully put some material between the cold pack and the scrotum so you don't get a cold burn. But put something there, but keep the scrotal tissue cool. Keep it cold to cool, because heat exposure can really mutate and disrupt the developing sperm, and it can kill sperm. And so again, that would be for an entire 90 days leading up to your attempts to conceive. Again, we'll get into more tools later, but a number of people also I've probably heard of the boxers versus briefs controversy. I guess it is, or whether or not people call it going commando with no underwear of any kind, boxers, briefs, or otherwise. Rather, turns out that the data on that point to the fact that there isn't really a big difference in terms of sperm quality if people wear boxers or briefs or don't wear anything under their jeans or shorts at all. The scrotum has the ability to move the testicles far enough away from the body in order to achieve lower temperatures if it needs to. It achieves that through a muscle called the cremaster muscle, which is a really interesting muscle, believe it or not. I was reading up on the biology of the cremaster muscle, something I never thought I'd spend too much time on, but that I ended up spending far too much time reading up about. And it's really fascinating. What you have is a muscle that's a smooth muscle tissue, unlike skeletal muscle, which is striated muscle, that is temperature dependent. So it has certain nerve endings and it has certain receptors on it that allow it to respond to local temperature and then to relax in order to essentially let the testicles to descend further from the body, or to contract and bring the testicles closer to the body in order to try and maintain the optimal temperature range. And it turns out the cremaster muscle can achieve that whether or not people are wearing boxers or briefs, although it stands to reason that any kind of there's no other name for it, undergarments, I don't know why that word is it just seems kind of antiquated, but undergarment that allows some movement of the scrotum and the testicles should be sufficient to allow these temperature variations to occur and keep things in range. That said, a little bit later, we'll go into some detail, really, because it's important as to why, for instance, if you are somebody who has big thighs, believe it or not, that it actually can lower sperm count substantially. Whether or not the big thighs occur because you're very muscular, the big thighs occur because you are overweight. It can increase the temperature. If you're sitting a lot, increases scrotal temperature for sure. And there are some other things that can increase scrotal temperature. Seat heaters in cars, for instance, terrible idea if you're hoping to conceive in the near future. And again, hot tub, things of that sort. Okay, so temperature modulation of spermatogenesis and sperm quality and function is key. That relates a little bit more to tools, but what happens? How does the actual sperm develop? Well, contained within the testicle, you have the cells, the so-called spermatogonia, which differentiate into so-called spermatocytes. You don't have to remember all this. And the spermatocytes undergo this process of meiosis. Meiosis is a form of cell division, which reduces the chromosome number to those 23 individual strands as opposed to pairs, right? So it makes them haploid as opposed to diploid. Very, very important for reasons that we talked about earlier. And the meiosis process in these primordial sperm cells, these immature sperm cells, is similar to the meiosis process that occurs in eggs when the chromosomes segregate, in that it involves these spindle-like structures within the cell. Now, why do I keep bringing up the spindles? Well, it turns out that the function of the spindle in the egg and the sperm is heavily dependent on mitochondrial function. And later, when we get into tools for improving egg and sperm quality, you're going to hear about a lot of tools for improving mitochondria. And it's not just because the mitochondria are involved in energy-demanding aspects of cell biology, but it's also because the mitochondria in this context are very, very important for the removal of, or the separation of, one set of chromosomes to give you these two sets of haploid cells, the egg and the sperm. And this is so important because many failures at fertilization, many failures at implantation, many, many miscarriages, and many birth defects that do survive after birth that are very detrimental, such as trisomies and things like that, occur because the spindles don't effectively pull apart the chromosomes in typically the egg, but it can also occur in the sperm. Okay, so these spindles and the fact that mitochondria are rich on the spindle are very important for generating these haploid sperm, again, 23 individual strands of chromosomes. That's occurring inside of the testes. Okay, so there's not as much long distance migration of the spermatocytes and the sperm cells as there is the egg, just when you think about the overall architecture of the uterus and the fallopian tubes compared to the testicles, but there's still a lot of movement. So within the testicle, if you were to look at the testicle in cross-section, and I prefer to call it that rather than cut the testicle in half, anytime you talk about anatomy, you actually talk about slicing things. That's what you would do with a cadaver, is what I teach and we do in my laboratory and frankly, in biological laboratories all over the place. But when you talk about it, you talk about if you were to take a visual cross-section through the testicle, what you would find is that there are a lot of different little tubes, a lot of ducts, D-U-C-T-S, TS, ducts. Those are pathways. And the main